And then what we have for the 16th is an article quiz. And that will do with the end of this chapter. And so we have two articles. One will be the article on the Miller experiment, and one will be an article on drug interaction. If you actually take a look at the quiz itself, I'm going to look at it. Continue the last preview. All right. Did the Miller experiment give enough detail to convince you that amino acids that are incorporated in life were created in the mechanism that he describes? I'll show that in just a moment. And is the use of racemic mixtures or individual enantiomers more advantageous in drug development? And so those are the questions that we're looking at, and they interact each one with one of the articles. Now, the first article, kind of interesting in that articles are kind of complicated, especially if you read a modern article. Although this article, dated 1972, actually is probably more readable than most. And so the Miller experiment, very famous experiment. Have you heard about that before? The Miller experiment? Okay. Yeah, I've heard the name. Do you know what it is about? Okay. My last slide in this chapter, we're going to take a look at that. But basically what it is this. Took a glass jar. Inside the jar, they put methane. They put a little bit of ammonia. They put some water. And they started sparking electrical sparks inside that mixture to represent a very ancient atmosphere with lightning. At the end, after they ran it, they found, also in there was CO2, they found amino acids. And so, this is how amino acids were made in life. And so, this is the actual article. A lot of people and a lot of older type of textbooks will, especially in high school, will prove that this is how life began. This is where amino acids actually occurred. Now, when you read a textbook, that is someone's interpretation of what the research says, and so this is the actual article that published that information done in 1972. I want you to take a look at it and read it and get a sense of what he thinks about the data. And a lot of textbooks say, this is the way. See if Miller's article states that this is the way. Okay, does he believe that? Okay. The other one is actually an overview article on drug action. A little bit less technical, but it does talk about drugs and having racemic mixtures. So go ahead and take a look at this one. It all goes back to reviewing stereoisomers, and so answer the two questions. Now, I don't want a book. Okay, I want you to answer the question. Don't go too short and don't go too long. Too short, not enough information, take up points. Too long, too much information, take up points. Right in the middle. Okay, this shouldn't be a page. This should be 4 or 5 sentences at the max about your understanding. I want you to engage the material, but not have to write a book on it. All right. Let me bring up an old school lecture. All right. Keynotes. All right. Let's get to where we were. We were right here. All righty. So, information transfer from the DNA all the way through to protein. In a lot of ways, that information transfer, we talk about it being a single flow, but there's a lot more things that actually happen. But in general, we talk about this: DNA to messenger RNA to protein. That is how genetic flow occurs quite often within our bodies to form function. And so that's what we focus on, although we now know we can take messenger RNA, go back to DNA. DNA does its own replication. We do make other RNAs for function, and so there's a lot more varied processes, but standard dogma DNA, messenger RNA, protein is what most people look at, represented here. And so the DNA double helical in nature, the messenger RNA single stranded, and it is a complement to the DNA. And the messenger RNA will make a complementary copy of what's called the template strand. And that messenger RNA is then processed. And once that messenger RNA is processed and shunted from the nucleus into the cytosol, we can then, at the ribosome, create protein. And we have protein. Very simple, very standard set of ideas. Nothing new, particularly for you in this. Now, we do want to talk about the mutations, a change in the genome. All right. And so if by chance we change one letter in the DNA, there is a possibility that an amino acid will change. Okay? It is not an inevitability. You could actually make a change and actually end up with the same amino acid because we have codons for each amino acid that are, we have multiple codons for each amino acid, but every so often mutation can cause a change. And in this case, this change is significant or not significant. Why is this one significant?
This is its first position.
It changes an amino acid, right? From one that is, yeah, it's the change in charge, isn't it? And it is in a very important site called the catalytic site. And so I need in a catalytic site originally in what's called the wild type, the original, a negative charge there. And that negative charge is going to attract to something and stabilize something or make an attraction. Well, if you change it from negative to positive, that fundamentally changes the lining of that pocket. And so what might have bound there initially that is attracted to a negative, now you have a positive to it, you would probably think it's going to repel. And so that change is really, really bad if it's there. But a change of a negative to a positive to an amino acid that may interact with water somewhere else in the protein, that might not be a bad change. It is a polar molecule interacting with water and a polar molecule interacting with water. And so just because we have a change doesn't mean it's necessarily bad. It's where that change is. All right, and so there happens to be errors all the time. But there are two kinds of cell, germline and somatic cells. Which one would have a greater effect if we actually incorporated a mutation? Germline, okay? Because the germline is fundamentally what is transferred from person to person or as we go through. A somatic cell has a certain amount of life, okay? You can have a cell that will be alive and functioning, and if it has a mutation for that one cell, it may or may not get copied, and it has a finite lifetime. But a germ cell is what's propagated into the permanent transfer of information. Now, we have mutations all the time. Most of the time they're in somatic cells, and so it isn't as bad. But if you do get a mutation in the germline, that will propagate. Now, mutations can occur, and they can be good, bad, indifferent. And there are different portions within our genome where we have what are called coding portions and non-coding portions, all right, or the genes or where there are no genes. If we take a look, if you mess up or you have a mutation in a non-coding area, probably has a much less consequence to the overall structure. But if you have a change in the gene itself, that could be an issue. All right, genes will accumulate changes over time, all right, and have the possibility of doing that. And so we can have multiple changes, although the non-coding portions we're actually finding have a lot of function. And so changes there can mess things up every so often. Now, we're going to find quite often that there are certain proteins that do certain functions. Okay? There are proteins that will break a peptide bond. All right? And so what we have to do is break an amide bond. And there is a certain orientation of amino acids that are positioned perfectly to break the amide bond. Now, we're going to see that we're going to study one particular protein that will break other proteins at the amide bond, and it has the perfect what's called the catalytic triad. It's very important. And it will bind to other proteins, this enzyme, and it will only bind where there are hydrophobic amino acids and break right there. Okay. Well, there are other proteins that have the catalytic triad that are perfectly arranged, and if you actually look at the sequence between one enzyme and the second enzyme, they're really, really similar. There are a few changes. The second enzyme will only break proteins where they are positively charged amino acids. The other one broke where they were hydrophobic. Now, these two proteins could be very related, meaning that the gene was developed for one of the optimizations to break a peptide bond, let's say at the hydrophobic ones. That gene got copied and got mutated a little bit. Okay? But the mutation didn't do anything with the reaction. The mutation had everything to do with specificity. Okay? This duplication of genes that had a little bit of change can be called a paralog. Okay? They had a common ancestry. They were the same protein that was duplicated, and the mutations changed their specificity, but essentially they're the same. We find paralogs in our bodies. We can also have orthologs, which are genes that are very similar between species. Here is an example, okay, down here where we have a gene and there are several different versions of this gene. And if you take a look at the sequencing, and this is representative of amino acids and the single letter designates, you may or may not know this. You can always look it up in a chart. We'll cover this in a couple chapters. But suffice it to say, each letter represents a different type of amino acid. And if you take a look at bacteria and yeast and worm and fly and humans, you would say that they should be really, really different, each one of those, right? Okay? Hopefully, a worm and a human we don't share a lot of similarities in look, right? Nor function. But in this one gene, if you take a look, between all of these, there is stars, and there are little dots, and then there's nothing. A star in what's called a consensus sequence means that this is conserved. This region right here, doesn't matter which gene you looked at, is absolutely the same. Usually conserved amino acids are where important amino acids reside. You can see something like this double dot, meaning that, okay, the amino acids are not conserved, okay, VL, VII, but what happens is all of these are hydrophobic. They are different, but they're all have the same property in terms of being hydrophobic. If you have nothing, then you can have all sorts of different views. So here we have a negatively charged, there we have a positively charged, this one's all over the place. And so that one doesn't have any commonalities between, but we can see that whatever this particular gene is, they are copies of each other because there is a lot of similarities between those genes. All right? This might be considered a very old gene because we believe in the development of life, which one came first of these groups? Bacteria. And this gene was important enough to be handed down through the species. Okay? Another interpretation, this gene is very important, and you see design in this. Okay? Data's there. They're all the same gene. They are considered orthologs. How you interpret it is a whole different ball of wax. Okay, and so in this could be a homolog gene where we think it might be an ancient gene that is used throughout all of life. Okay. Oh my gosh. I thought I would never see this past Dr. Krebs, right? Delta G, delta H, T, delta S. Oh my gosh, what are these? What is delta G? Radiant gives energy or change in energy. What's delta H? Enthalpy. What is delta S? Entropy. Okay. All right. What's T? Temperature. Good. All right. So, what is enthalpy? Do you know? We've used enthalpy. We use delta G for spontaneity. Delta H is not necessarily spontaneity. Okay. Enthalpy, when we first learned it, okay, is related to energy. You can think of it this way. Delta H is the potential bond energy. Okay. And remember those problems where we calculated delta H by looking at all the bonds broken minus all the bonds made type of things? This is kind of a measure of how much potential energy is in each bond that is going to be broken. Okay. Delta S, entropy, is the measure of disorder. And we like disorder. That's what we tend to do if we want to go spontaneously. And then delta G is a measure of whether the products at the end overall are, what their energy is compared to the reactants. And for things wanting to go spontaneously, we want delta G to be a negative. Okay? And so all of these are important when we think of biochemistry and what must happen. Okay? And so in this case, if we go from the synthesis of a macromolecule from monomers, we are going to mess with entropy. Because there's order when we make the macromolecule where there is disorder with the individuals. So we have to overcome that with, when we break the bonds, we have to give up enough energy so that this becomes an overall negative process. All right. Now, in the synthesis of proteins, what we need are amino acids, and amino acids are synthesized from basic carbon, hydrogen, nitrogen. This is the experiment that was done by Miller, and what they did was they made an assumption of what the early atmosphere of the planet was. And this is before we get bacteria that created the oxygen atmosphere. Okay? And so we had no oxygen until there were bacteria. We had no bacteria until there were amino acids and proteins. And so they believed that there was nitrogen, methane, hydrogen, water in there. And they used electricity. After a long period of time, they isolated what was in the water, and they found amino acids. Okay? Now, it'd be interesting to look what kind of amino acids they found. Okay, that's important. And also, what we find in this reaction is a racemic mixture of amino acids. When you look at the human body, do we see a mixture of the isomers of amino acids in our body? You know. No, we only see one version. We see the L version versus a mixture of L and D. All right. This reaction is going to give you L and D versions, or you can also call them S and R. For a long period of time, this was a interesting argument from a Christian perspective, in that, if we synthesized all our amino acids this way, why would we only incorporate L versions and not L and D, since we don't find any D amino acids in biological systems? So this was a wonderful Christian argument for a very long time. What would detract from the Christian argument about, oh, only L amino acids are found in biological systems. What if you found something that had a D incorporated into it? Uh-oh. And unfortunately, we have now discovered some bacteria and viruses that have D versions. So we can't use this argument as a sole argument of why life was created and not synthesized this way. But look and see how Miller describes this experiment, because in so many texts, this is the definitive, this is how it is, argument. I want you to see how Miller approaches this. Now, Miller is not a Christian. He is an atheist. And so he doesn't come from any particular perspective that says it has to be this way. But it's interesting how he conveys the information. All right, chapter 2. Do a little bit more GChem-2, and then we'll talk about water and membranes. And so what's important about what we look at, and especially as we go through metabolism, everything's driven by energy. Most of our, or all of the reactions in our body have to either derive energy from someplace and or convert energy. And so what we totally understand is the energy from the earth that we use ultimately comes from the sun through the thermochemistry, the nuclear chemistry that happens, the fusion that happens that gives off that energy. That energy is trapped in our ecosystem mainly by photosynthesis and carbon fixation, of which those are the molecules that we use for energy derivation within our bodies. And so the potential energy sources that we have are molecules that can be oxidized, okay, in a redox reaction. We also will see electron carriers, which are energy molecules. We will then convert these into gradients of ions. You get an electrochemical potential. And ultimately, we will get energy in the form of a physical molecule, ATP being the most obvious, although there are other triphosphates that can be used. The interconversion, you have a catabolic and an anabolic process. The catabolic is breaking things down to make energy molecules. Ultimately, we refer to that as ATP. And then the anabolic is the synthetic process by which we make more organized molecules or larger molecules using the energy we have derived. And so we will spend a lot of time on the catabolic process in metabolism, and then we'll shift our sense. And right now I am teaching in Biochem 3 or molecular biology the anabolic synthesis. So we spend a lot of time breaking things down and then building them back up. Back to thermochemistry, generalized reaction where you have products and reactants and you have the coefficients. We can find equilibrium. Equilibriums drive every reaction almost exclusively in biochemistry. There are very few reactions that are not in equilibrium. And so this is an important concept that we must understand. When not at equilibrium, the delta G will tell us about the tendency to go towards product. And you learned of a delta G 0 or a delta G naught as the standard free energy. Okay, the standard free energy is at 25 degrees, 1 molar concentrations for all reactants and products in the equation, and 1 atmosphere for gases. That is not enough for biochemistry. Biochemistry has a couple other things. What does it add to make a standard? pH, because everything is revolved around pH in the body. What else? Water. Ooh, water concentration is 55.5 molar. What else? Magnesium. Magnesium is very important. I'll show you exactly why in a moment. Uh-oh. Let's do a GChem question. How in the world do you get 55.5 molar for water. Have you ever seen the molarity of water before? You haven't, have you? Okay. How can you mathematically calculate the molarity is 55.5? I told you it was that. Could you actually calculate that? Okay, first of all, what is molarity? What was that? What over what?
Grams per liter.
Not grams per liter. Not molarity. Moles over liters. Okay, so you got to get moles over liters. All right. You don't know that about water, but you know a lot about water. You know a lot of constants about water. Okay. What constant has a volume that you know with water? Okay. What do you know about water? It boils at 100 degrees Celsius. It freezes at 0. You got that right. Its heat capacity is what? Oh, come on. 4.184 joule per gram per degree Celsius, right? You know that. What is its density? 1. Okay, great. You're half right. That means you're half wrong. 1 what? Gram per milliliter. Okay, great. Now, I got a volume here, I got a volume there. Can I change milliliters to liters? I can you? 1 times 10 to the -3, good. All right, I'm there almost, right? I've got liters, grams over liters. That's not what molarity is. What one more multiplication can I do to get this done? The molar mass of water. How many grams make a mole of water? 1 mole weighs 18.02 on CHEM-321, which is always right. Okay. And you know what happens if you do that? Moles per liter or more. So you could have done that. But by me doing it, that's magic, right? Okay? You could have got there. Although it looks complex, it's just you can win. All right? And so the molarity of water is important. We'll see that in just a moment. So we assume everything's pH of 7. We assume everything has magnesium in it. We assume there's 1 molar concentration for everything, and we assume it's 25 degrees Celsius. Okay? If this biochemistry is happening in your body, what one thing is wrong on there all of a sudden? For the standard. Temperature. What's our temperature? In degrees Celsius? 37. Okay, so delta G's that are standard are not real. That happen in the body. All of a sudden, we're going to see that it's not real in a lot of different ways. But every estimation that we make will be derived from the delta G because all standards can be compared. All right. The standard energy can be calculated using negative RT, natural log of the equilibrium constant, and we can do mathematical calculations like that. All right. There's a book example of this reaction. It shows the equilibrium. You could plug and chug those things in, okay, given the constants. I don't think that none of you would not be able to do that. If I gave you R, if I gave you T and I gave you the equilibrium, all right, you should be able to do that. And so when you take a look at the kinds of reactions that we have, if you assume every concentration was 1 molar, what would actually happen? Would it drive towards product or if it would drive towards reactant? We can see lots and lots of types of energies that are given off. kcals is an older numerical value. You may see them in old journal articles. Kilojoules is a more modern way that we do this. And we can see the most important one, or the reference one, as this one right here, negative 30.5 kilojoules per mole. Why is this the important one? Why is this the reference one? What are we breaking down? ATP. We're taking only one phosphate off. All right. Almost every reaction in the body can be funded, or meaning that all of these can be run in reverse if you break this down. Okay? But to make ATP, you have to have a few reactions that are in higher energy so that I can make ATP. So almost all the reactions that are above 30.5 are involved in making the ATP at some point in time. But this one is the important one when we take a look at this. Now, the standard free energy, okay, we already said it's not 25, it's 37. Also, the standard free energy means that there were 1 molar concentrations in everything. Very rarely, if I even know one single concentration in biochemistry that gets up to 1 molar. I don't believe so. If you get up to millimolar concentrations, that is huge in a biological system. A lot of times things are down in micromolars or nanomolars. All right, so 1 molar is way too concentrated. So there's issues with that. And so if the concentrations are not at 1 molar, then we get a ratio, okay? And the concentrations in this case can change the direction. If the ratio of the products over the reactants is below 1, then when you take a negative log of something that is less than 1, that can influence a reaction. If something has a delta G that is positive, if by chance you change the concentrations large enough, you can make the concentration difference enough energy to drive the reaction forward. And so the body uses changing the concentration between products and reactants to facilitate things towards running to product. And so that is an important aspect. So what we have to do is really look at the concentrations that are really there. And what we do is we modify the standard versus the reality of the situation, the concentration difference, and we can get a delta G that is negative, even if the delta prime 0 or prime naught is positive, if we manipulate the system. All calculations in delta G and delta G prime 0 are theoretical maximums. And even though we could get negative delta G's, it doesn't address the activation energy. And so even though it wants to spontaneously go, that doesn't necessarily mean it can get there if the activation energy is too high. This is why we use enzymes, is to lower that activation energy so we can get to equilibrium faster. Ooh, that is interesting. Okay, we're going to see it again. Here is the application of Hess's law, and we've seen this before. Reaction 1 and reaction 2, if we add them together, can proceed, and quite often we use the ATP value of negative 30.5 to drive reactions. Strange. Okay. Again, the energy molecules that are important to us, molecules that can be oxidized. This is basically food. What are the 3 kinds of food you can eat? 3 classes. Proteins. Carbohydrates or sugars, and fat. Okay? All of those have a potential oxidizable amount of carbons that are coming in. They can also contain sulfurs and nitrogens, and we have to watch out for those. The electron carriers, you've probably seen these before. FADH2, we'll look at the structures a little bit later. NADH and that's supposed to be NADPH, not HAE. That's funny. That is. And magically it changes. NADPH. Gradients. A lot of times we'll talk about gradients of sodium or gradients of proton. Those are probably the most often used, although those are not the only ions that can create a gradient. And then the chemical energy, ATP. Ooh, we've already talked about this. We said the energy of ATP resides. Why? There was a concentration of what really close together? The negative charges. That is one aspect of why there's a lot of energy in ATP. Here's the second aspect. Once the bond is broken, the phosphate creates more resonance structures. Resonance is a huge amount of energy. And so once that last phosphate is released, the increased number of resonance structures also drives stability. So those are the two areas in which why ATP has lots of energy. Okay. Now here's the problem. Remember that concentration standard for biochemistry. It had pH, it had temperature, it had water, it also had magnesium in it, right? Okay. Look at what magnesium is doing to ATP. What does the magnesium coordinate to? The negatives. Does that increase ATP's energy value, or does that decrease ATP's energy value? It decreases it. Because of the presence of the magnesium, in reality, it stabilizes that negative charge, which doesn't like to be there, and actually reduces the amount of energy. But remember, the delta G that we use that says negative 30.5 kilojoules is based upon 1 molar concentrations. Look at the concentrations in the cell. And these are in different tissue types. Concentrations, are they in molar? They're in what? Millimolar. And they're very small fractions of millimolar. That tends to make the energy value for ATP higher. Okay? And so in real life, most of the time ATP has more than 30.5 if you actually do the real calculation of the concentrations and the magnesium and everything in there. And it gives off more energy. But here's the problem. Every reaction that you're referencing to also has different concentrations, and they would require more energy. Even though ATP is giving off more energy, each reaction requires more energy, and so you would be doing a lot of individual calculations and doing the subtractions in terms of Hess's law. And you know what? If you just add up the standard reactions, you get about the same number. And so we don't do all the individual calculations, and we just use the standard, and 99% of the time it comes out what we expect. If you did something and you got 99% of the right every time, would you use that technique on the exam? Yeah, if you get a 99 on every exam, at least I would say 95% of you would be happy. I know there'd be 5% in this room that 99 is not enough. Okay, but you know, if something doesn't work out, you can always go back and really work it out and see why it didn't work out. But we use the standard energies, even though we understand they are reference points and they're not the real values. They're close enough for us to do calculations. ATP, I love this. Described as thermodynamically unstable, but kinetically stable. Ooh, what does that mean? I'm using stable, right? Got that right? Both of them. Unstable and stable. Okay. What in the world does that mean? What is thermodynamically unstable?
It's ready to react.
Yeah, it wants to react. It wants to give off a big negative value. It will release tons of energy if you just break it apart. It wants to go. Okay? Kinetically stable. What does that mean, though?
Is it the same?
Okay, so if I put ATP in water, it's going to give off lots of energy to break apart. And all I need is water to break that phosphate. But if I put it in water, does ATP break down? The rate at which it converts by itself is infinitesimally small. The rate at which it moves from one to the next version, from the triphosphate into the broken pieces, doesn't happen at all. It's slow. Unless you have an enzyme. What that means is it has a very high activation energy that is not overcome by the physiological concentrations and temperatures. And so you can have ATP floating around, and even though it will give off lots of energy if it broke down, it just doesn't go through the reaction. It is stable because it has that high activation energy. And so it can float around as a high energy molecule without just breaking down automatically. Okay? So I can oxidize things, and so electron flows can actually be calculated, and you've done this in CHEM-2 using the Nernst equation to actually calculate how much energy is given off. And so when you do this, you can get the standard reduction potential, and using this equation in number of electrons, Faraday's constant, which I would give to you, and the delta E, the change in the standard reduction potential, you can calculate delta G. A very simple example of that, and unfortunately, that registered trademark is actually supposed to be an arrow. I don't know why I changed it. The provate plus NADH plus a proton will produce the molecule lactate plus NAD plus. 2 electrons flow from this NADH into provate, and I'm missing 2 electrons from this, but I gain 2 electrons right here. Here are the half reactions, and if I take acceptor minus donor, I get a numerical value. I can place it into this small equation. Here's the Faraday's constant, which I would give you to use, and you can calculate that this reaction would be spontaneous because it has a delta value, a negative delta G for the overall process. The one big thing that you have to watch out for, and most people miss on this kind of question, is the flow of electrons. In this case, are 2, almost exclusively in metabolism. We are moving 2 electrons in the form of a hydride. So the N is 2. Okay, if I give you the electron potential for the acceptor and donor, and I give you the Faraday's constant, again, I don't foresee that that would be a problem for you to calculate that. And so we are looking at lots and lots of energies. The last concept, or the next concept that we really look at is water, like you haven't seen water before. Okay, water, tetrahedral, and its electron pair geometry. It is bent in its molecular geometry. It has 2 lone pairs of electrons, and it has a potential to make hydrogen bonding. How many hydrogen bonds can each, oh, and it's right there. Each water can make 4 hydrogen bonds, so it is very, very energetic and favorable. But here's what I want to show you. Here's the energy for the covalent bond. Oxygen, hydrogen. Look at that number. Huge, right? We say hydrogen bonds are huge, but that's an intermolecular force. The stabilization is only 20 kilojoules per mole. Intermolecular forces compared to covalent bonds are much, much smaller. But if you take a look at hydrophobic interactions, you may be talking 3 or 4 kilojoules per mole. And so they're all relative. This is relatively a very strong interaction. All right, and so a lot of things are driven by that hydrogen bond that water actually uses. All right, we have come to the end of the time.